So we are going to continue with the process of fertilization in the human. Now, in the course of fertilization, as you know, that the sperms are going to come and bind with the ovum, and then penetrate inside, finally dispensing the entire sperm nuclei inside the ovum, and also along with it there will be the proximal centriole that will also go inside. Now for that particular process, what we are going to find is that there is an distinct interaction is going to take place. If you remember the diagram of the ovum, then around the ovum, if you remember, there are large the cells are actually that are present called the corona radiata, and the corona radiata, it actually through that corona radiata the sperm actually penetrate inside, all right, and then finally it will reach the zona pellucida layer. Once it reach the zona pellucida layer, right there will be an interaction taking place between the anti-fertilizing and the fertilizing of the sperm and the ovum. Now when that happens, at that particular point of time, the acrosome or the head of the sperm will actually break open, or it will rupture, releasing all the hydrolytic enzymes that are present inside. Now, if you remember, it actually has got many type of enzymes: the hyaluronidase enzyme, the proteases enzyme, different varieties of proteases enzyme. Also, there is something called the zonalysin that is also present. Now, the zonalysin is responsible for penetration or actually dissolving the this zona pellucida layer. Now, once it will keep on penetrating that, it will move further down inside and will reach this perivitallin space. All right, this is the the last layer we can say that is there. Now on this, once it reach that particular region, there are these cortical granules that are arranged around it. Okay, means actually there are two types of things that will be present. One is the protein receptors on the surface, and then finally the cortical granules that are there inside. Now the moment the sperm actually reach this particular portion, what will happen is it will the sperm head will actually make a little twisted turn, we can say, or actually it's actually that particular point of contact. The plasma membrane of the sperm and the plasma membrane of the egg, they fuse together, right? Okay, and the moment they fuse together, the sperm nucleus and the proximal centriole will actually be released from it and will get inside the main egg cell, right? Now this is the egg cytoplasm, if you remember, with the nucleus present inside, the haploid nucleus present inside. The sperm nucleus also will enter right there. But the moment this entry takes place, at that very moment, the cortical granules also will fuse, means both the fusion will take place simultaneously. So the cortical granule will also fuse. The moment this cortical granule fuse, it actually release all its entire content, the cortical granules that are there present inside. This entire thing will be now dispensed into the this perivitallin space. This interaction, or this fusion of the cortical granules with the egg plasma membrane, which release this all these cortical granules in the perivitallin space, will actually make the entire vitallin layer impenetrable for any other sperm. Because if you remember, even I can show you the picture one more time. Here you can see there are numerous sperms are adhered to the surface of the ovum, but only one will be allowed to enter, because the moment one is able to enter inside and then break inside the perivitallin space, even like it's actually vitallin membrane, once it fuse with the vitallin membrane, that very moment this release of this cortical granules will actually make the rest other nuclei, means rest of the sperm impenetrable, means incapable rather to penetrate this perivitallin space. Otherwise, there will be a single ovum fertilized by multiple sperm that can happen, isn't it? And that is definitely not accepted. So that's why this is actually a way of preventing that from happening, preventing any multi-fertilization event or anything like that. So this is how the sperm actually release its nuclei inside. Now sorry, nucleus inside rather. Now once that happens, definitely the fusion of the two nuclei will take place. But if you remember, it is this particular point of time that the egg cell will now enter into a stage of completing its meiosis 2 division, finally releasing the second polar body, and then the actual division or further like mitotic division as it will normally take place will actually commence right that at that particular point of time. So now let's learn about the places where it actually it takes place. If you remember, the ovary is actually like held by this fallopian tube, right there, and the ovum is released from there, isn't it? So once this ovum is released, it actually stays in the infundibulum region, and then it will be moved backward towards this isthmus junction, right? This is the ampulla and this is the isthmus, so it actually starts moving towards that side. And in that particular ampulla isthmus junction point is where the fertilization mainly takes place, because the sperm is traveling from here down here and then finally will reach in this particular isthmus region. Once it reaches there, the egg will be found present, and there the fertilization will take place. The completion of the meiosis will be occurred, like it will all happen and everything, and then finally the zygote will be formed, which is in the stage where now the zygote will now divide. See, it's written as pronuclear migration because they are separate. The release of the second polar body will take place right there, and then finally the main fusion will take place, finally initiating the first meiotic or mitotic division, sorry, first mitotic division. Now this first mitotic division will produce the two cell structure of the zygote. I mean, it's actually the stage where the zygote has got converted into the first embryo, alright, the two cell structure. This conversion is what is known as the step of cleavage formation, the first cleavage, then it followed by the second cleavage, and thus the further division will take place. Now this entire thing to happen, it actually takes somewhat around 24 to 36 hours to complete itself, and then finally it will just continue with the entire process and it will keep on going further, right? Now that entire period of time, the zygote that has been formed here, the zygote will be pushed outside of the fallopian tube into the main region of the uterus. This is all mediated by the ciliary like epithelium that is present all around this area. Ciliated epithelium that is there, it will actually push the entire thing slowly outside, and in a course of 7 to 10 days time, it will actually come out and will fall into this uterine wall, where it will actually now fuse with the uterine wall. Okay, so this formation, this entire division and its formation is what is like known as the embryonic development, and it actually like starts from day 1 and it continue further down up to, see here it is day 7, right? Day 1 fertilization, from there the first cleavage, it will take entire 1 day to complete the fertilization. Then from there, day 2 the cleavage will actually, well, it is shown like multiple cells are there, but in reality actually 2 cell to 4 cell structure actually is achieved by day 2. And then the finally the process of the compaction will take place. Well, compaction stage is right there where the modular formation will take place. I think it is exaggerated slightly. No, fine. No, it's all okay. Yeah. So the first cleavage and the second cleavage stage, it will take place right there. I'm sorry, I'm not sure about the third, like this stage will happen or not, but the compaction and the morula formation, the morula formation actually it will take place in the end of day 3, and then on day 4 the main differentiation will take place. All right, differentiation is actually a stage where the entire cell will be divided into now the outer ectoderm and the inner endoderm, and then finally the mesoderm layer will be created later on. So actually this will be discussed in detail in another video to you people. Now finally, when the cavitation and all these things will happen, finally on day 7 the entire zona lysine will now leave, because the zona pellucida will actually leave, because the zona pellucida was actually covering the entire structure up to this particular point of time, and in that particular point of time it will release itself, thus allowing the entire thing to go and now penetrate or fuse with the wall of the uterus. Okay, and then the further development will take place. This all will be discussed in a later video. So this is actually the step of fertilization that occurs in the female, and then in a course of this 7 days, the entire ovum and everything will now transform into the embryo and will now get fused with the wall of the uterus, called by the step of called implantation.